So thank you so much for coming. This is kind of the first talk in this exhibition. But yeah, I'll start with a bit of exhibition backstory. So I came to James with this kind of idea, my obsession this year to be about systems across scales. And we can ask these questions of whether there's any kind of common features between them. And so the pieces that I put together, this is one I did with James. We were trying to mimic biological processes, particularly things like growth. So this was looking at the kind of collective dynamics that go on inside cell growth, but magnified. And in the pieces that I put together, I looked at really two different kind of scales. So on the left, you see two different kinds of slightly fictitious insect nests that were 3D printed. And then also on the right, this is kind of a slice of leaf tissue that's trying to illustrate the kind of biochemical signaling that's going on between leaf cells. And to give you a bit of background, really, I love designing these kind of interactions, often with mixed reality and projection and things like this, to try and visualize these processes. But also before that, working with robotics and this kind of interaction. So this is a robotic camera rig that you can control with your hand. But more recently, I've been working on kind of building simulations, particularly of living systems, and using code to define these kind of complex shapes and patterns. So this kind of leads on this paradigm of parametric design, which means that we can have these algorithmic recipes for objects, and there's this tight mapping between code and geometry. So here you see a whole guitar that I've defined with a bunch of algorithms, or a bunch of code blocks. And what that means is that we can have this huge feature space of kind of customizable guitars. And what I realized in biology is actually this is the same thing, but even better. But there's this very tight mapping between code and geometry. So what we see is that genes, or particularly genetic code, are translated into these strings of amino acids or proteins. And these proteins then fold with unique fold into shapes with these unique functional properties. And even more than that, these proteins can then bind back to the DNA sequences to decide which programs or which other proteins get built. And this is a really powerful idea. But what it means is basically that in biology, information actually has a physical shape. And that physical structures can not only store information, but they can also process it and perform computation as well. And so I'll be talking a bit more about these kind of structures. So really the kind of paradigm or idea behind this talk, nesting automata, is about looking at biological software architecture and whether we can mimic it at all. So I'll be focusing on these kind of biological design patterns as if I'm kind of looking at it from the lens of a software engineer, and trying to ask these questions of how is it that we get these kind of complex recurring patterns of growth in nature. So this is a microscopic fern spore that will then form this fern frond. But we used to think that these shapes were just kind of down to things like fractals, but that's a very top-down view. Actually, what I want to try and get into in this talk is the bottom-up approach that leads to this kind of complexity. And so one of the things we can do more with these computational techniques is try and simulate life in order to understand it better. And this is exactly what kind of early computer scientists were thinking about. So Alan Turing was looking at the end of his life. Unfortunately, his work was cut short. But actually, he was looking at how things like zebra stripes emerge. So he was looking at how chemical patterns are used for communication and also for self-organization. And likewise, John von Neumann, actually, when he was at Los Alamos, he had access to computers, and he would try and build these simulations of self-replicating machines. And actually, he ended up forecasting a lot of the discoveries that Watson and Crick made to do with DNA. Because we can ask also, why is it that structures in nature are able to self-assemble and self-repair? Right? So there's a seed that can fit on the tip of my finger, and why is it that it's able to build this giant sequoia that is multiple stories high and can live over 3,000 years? But not only that, but it can adapt to changing environments and also withstand particularly adverse conditions like forest fires. And that's something that we want to copy, obviously, in the structures and kind of software that we build. And this is why I got interested in things like bio-inspired robotics. So how can we control, potentially, swarms of very simple robots that when we're put together, they can achieve these very complex functions? And even moving on from that, I've been thinking about how things like swarm robotics might apply to things like intelligent construction. So how can we build architecture that might even build itself? Because this is what I asked, I tried to get GPT to think about the future, and I didn't really like what it gave me. This is not how I conceive of it, and I think that's what I'm going to try and get into, but I don't think this is the future of architecture. So, if you remember your kind of GCSE biology, we have these levels of organization. And actually, if we take this kind of holistic view, which is what systems biology does, we can start with the level of kind of colonies or the superorganism, and then we can ask questions from behavioral ecology, like how do swarms of insects build particular nested structures? And below that, we have organisms, of course. Below that, organs, tissues, and then cells. And that's the level where we can ask things from cell and developmental biology, like how do plant leaves get their shape? And I'll come back to that one. And below that still, we have organelles and then biomolecules. And then we can ask questions from biochemistry, like how do proteins assemble these very intricate complexes, and then use big structures from that. But the kind of key idea I want to kind of drive home is that the cell is not a machine. We can no longer view it like that. It's actually an entire ecosystem in itself. And this is a kind of complex lens, rather than the reductionist approach we've taken in the past in genetics. So since the 1950s, the kind of marvel and the discoveries of DNA kind of made us a bit cocky. We thought we'd cracked the code, but actually it turns out there's a whole lot more going on than just genetics. And if you're like me, you didn't do anything past GCSE biology, you might think the cell is this kind of vacuous, fluid-filled sac. But actually, what I'm going to get into this talk is that there's a lot more going on in there. And what got me excited when I was doing more reading was that there's actually this entire cytoskeletal network, which is this network of constantly moving and shape-shifting tubes that not only support the cell, but allow us to transport important cargo where it needs to get to. So really, this is about trying to take new perspectives and look at things from a complex lens. And with that, we take an interdisciplinary approach. So we have the existing natural sciences, with biology, physics and chemistry, but then we also kind of butt onto that this meta toolkit of computer science, network science, and more recently, kind of complexity science. Because when we have problems like cancer growth, for example, this is one of the first things I looked at, you realize that they have very complex dynamics, and it's something we still really don't understand and we struggle to model, because the microenvironment really matters. And what that means is that the rate of growth is not at all predictable or can be easily modeled by simple functions. So for example, because of overcrowding in the center of a tumor, the cells are so squished that they grow slower than the cells on the borders. And so you get these very irregular, jagged edges where the cells are proliferating rapidly to try and escape this overcrowded area. And more than that, the cells themselves are actually smarter than we let on before. Cancer cells particularly can swim through tissue, and that's why you get metastasis. But also they can sense these mechanical stresses as well as just kind of communicating with biochemical signals. And I'll try and get into some of this as well. But yeah, if we kind of now think about cells being smart and think about this from a computational lens, perhaps we also need to then kind of redefine our conception of cognition and computation to fit this description. So in this talk, I really want computation to be seen not just as this mechanistic series of mathematical operations being done on silicon hardware, but actually just generic information processing that's agnostic to that. And equally, I think Krankovich has this great definition for cognition, and we can see that as this network of concurrent morphological or morphogenetic, meaning shape or growth related, computations that are unfolding as a result of self-assembly, self-organization of physical, chemical, and biological agents. And the kind of abstract model that we can take for all of these systems, hopefully, is this idea of interaction networks. Because that allows us to ask these questions of very disparate systems, like how is information processed locally and globally? How are the distinct parts arranged? How is the order maintained? But also how are group decisions made? So how do these kind of little parts achieve some kind of consensus? So this is me trying to use that idea to organize the ideas for the talk, but it didn't fully work. So the first kind of behavior I'll be talking about that is common across these systems is self-organization, and that's really important. And it's this kind of slightly wacky idea or wishy-washy idea, but it says that basically spontaneous global order can arise from these very minute local interactions, often in great numbers. And in tissues, on the level of tissues, this was inside the cell, right? So what we see during cell division is that the chromosomes will actually line up very neatly and be pulled apart by these spindles to divide the kind of genetic material equally between the cells. And in tissues, we also see kind of similar intelligence, where the cells will know how to divide and organize into distinct zones by themselves that will help us kind of mark different areas of tissue. So for example, one bit's going to become the leg, one bit's going to become the head, and that's done in a predictable fashion, so we don't end up with organisms with, most of the time, kind of organs in the wrong place. And the other kind of important collective behavior that I want to talk about is synchronization. And that's the way that these interoperating agents are able to kind of reach a coherent or kind of consistent global state or some kind of consensus. And one of my favorite examples of this is firefly synchronization. So when fireflies are mating, they flash their tails in order to attract mates. But you can imagine that in the forest this is quite dark, it's quite hard to find mates. And they are competing, but they decide to work together. Because what they can do is, if they all flash together, it's more likely that other fireflies will be able to find them. And so the way they do this, especially in kind of a bush, you can imagine they can only see their closest neighbours. But if they just keep flashing and doing their own thing, but they just pay attention to their nearest neighbours, what we find is just, even with this very limited communication, they're able to all eventually sync up to make this huge kind of flashing bush that attracts mates. And what we find is actually this can be modelled very nicely by something from physics called coupled oscillators. And this is seen both in kind of mechanical circuits like springs, but also electrical circuits like inductors and capacitors put together. And all you need to know really is that everything all of these things have somewhat a natural frequency of some kind, at which they prefer to vibrate. And when we are able to match this frequency, we get maximum energy transfer. And when we have many together, if they can kind of all operate at the same time, they tend to want to find this kind of coherent state between all of them, where they're matching the kind of frequencies they're naturally oscillating at. And in biology, we can talk about this because there is energy transfer going on between these system components that kind of chatter. But we can talk about how, even with limited communication, systems are able to reach a kind of synchronized state. So not just firefly mating, but also at a cellular level, plants have circadian rhythms, and they will sync up as kind of cells for synthetic activities. But also in synapses, in neurons in the brain. So the kind of first key point that kind of draws on what we were saying before, I want to kind of address is this idea that structures themselves are processing information. So you can think of the brain as this kind of high-dimensional guitar. That when we pluck, we get these kind of ripples of electrical activity moving outwards. And I'm getting looked at by the neuroscience in the room, so I'm going to rush over this part. But what we know is that biological structures themselves can amplify, filter, but also integrate these signals from the outside world. And what we know is that the kind of shape, the morphology, and the metabolic constraints, i.e. kind of energy that cells are using to process, those constraints drive the signal processing. So to give you an example from neuroscience, we have synaptic integration. So the neurons themselves in artificial intelligence, we tend to treat them as these kind of point approximations, which is completely wrong. They're actually this kind of distinct constellation of dendritic branches. They have their own unique shape. And the shape actually changes their kind of bioelectric properties. So what we see is that all of the synaptic inputs undergo this kind of series of logical operations, like a series of logic gates, that as they're all kind of collected, and then a decision is made whether the neuron should fire. And this means that this is a way of eliminating noise, is that if we get one input, it actually might not necessarily fire. But under the right conditions, for example, when both of these dendrites receive inputs, then we actually get an action potential and a spike. And so this is the first example where really a neuron, in computer science terms, is actually a whole neural network in itself. So structuring information is a really key part of intelligence on a more kind of cognitive level. Because what we're doing is really filtering these streams of constant streams and overwhelming streams of environmental input into these kind of coherent internal models that help us do useful things for our survival. So we can see attention as this kind of black box mechanism, but it's a compression scheme that basically stores only the important associations that are relevant to our survival and structures them neatly in the kind of configuration that helps us do stuff. So a great example of internal models, just to kind of drive home the point that it's also spatial, is place cells. So in mice, we can see when we put them through a maze, they'll wander around, but actually a particular part of their brain, the hippocampus, has these things called place cells, and they'll fire when mice are in particular locations in the maze, so they're linked in that way, a kind of internal map. And what we see is that only the configurations that help the mouse, the routes that help it get through the maze, those are the ones that get reinforced and played back during the mouse's sleep. So when they try and do the maze the next day, they remember the correct way to get through the maze. But even more, you might think plants are stupid, and this is another kind of intelligence completely, but actually they have a kind of memory, and this is getting into kind of epigenetics. So we find, for example, in holly bushes, that when they're browsed by deer, the bottom of the bush, the leaves will actually become more spiky. And this is because stress mechanism is getting activated, which is a bunch of kind of genetic switches that are getting flipped. And what that means is something called DNA methylation. But what that means is that even though these leaves have the same genetic code, the shape or the morphology goes completely different because these ones have been attacked by deer. So that leads nicely on to this kind of next point, that actually not all computation being done is neural. So other cell types can actually communicate through these kind of bioelectric signals. And actually even kind of dumber than that still, but some shapes, some materials, passively, just due to their properties, can process information. So we see that actually we have things like compliant mechanisms, where a material can have kind of a stable or latching different stable states in which they can latch between. So we have this kind of switch, binary switch effect. But to give you an example from biology, we have this thing called morphological computing, which is all it's saying is that shapes themselves are doing some kind of processing. So in the case of insects' legs, they're actually one of the smartest structures we know. Not only do they have this kind of very smart suspension system, but actually the feet are very good at sticking surfaces, but only under the right conditions. Because if you think about it, insects need to be able to walk up walls, so they need to have a lot of kind of adhesive force, but they also need to be able to pick up their own legs, otherwise they just get stuck in place. So what we see is that, for example, if an insect is walking up and then it's struck by a drop of water, if that was a reflex that kind of stopped the legs from, made them kind of grip on, it would be way too slow. So instead, what nature's decided to do is to use the structure itself to kind of store this or program this important behavior, and we program that behavior into the material. So through this combination of kind of hairs and a claw, we see that it's able to selectively shear. So this leg will only come up if the insect is kind of slowly prizing it off. Actually, if it feels sudden force, a bit like a seatbelt, it will grip even harder. And so we have this very clever kind of material that enables insects to stick and also not slip. If we look at something on a really small scale, we have ion channels. And this is actually a kind of protein structure that acts like a gate. So a bit like a transistor, it's very sensitive to certain inputs. So for example, this one, when it gets struck by light, a particular wavelength, the protein will actually change its shape. And it then will open the gate and allow these ions to flow. And a much bigger current can flow, which can do kind of useful processing. And so the kind of next key point I want to talk about, which is about when you put these things all together, is these collective dynamics. And the idea that the whole is greater than the sum of the parts, which again is this idea of complexity. So we see that complexity arises from simple parts that are interacting in great numbers. Even if we understand the parts, they can have unpredictable behavior. And especially, they're unpredictable if they're heterogeneous, so like cells. In the past, we've tried to use aggregate models in biology, and this is why things like mathematical biology fall down, in my opinion, because they try and generalize the idea that everything, all the kind of component parts are the same, and that's just not the case. So instead, we can use these discrete and agent-based models rather than continuous approximations. But what we see is that when you have kind of frequent concurrent interactions, you get these strong interdependencies within the system. And this is what sets up these feedback loops, which leads to nonlinear or chaotic behavior. And if you've heard of the butterfly effect, it's exactly that. It's the idea that these tiny kind of interactions between parts can sometimes have very big consequences, and that actually does a lot of useful stuff in biology. So if that made no sense, here's a cool picture. This is bees collectively doing this kind of shimmering as a defense mechanism. So individual bees have this behavior where if you piss them off, they will stand up on their rear as a kind of way to ward you off. But when you put them all together, this individual behavior becomes actually a much more powerful and potent aggressive signal to piss off that actually is just like a kind of Mexican wave. But as we see, this is a kind of example where as a group, they're able to kind of achieve a function that is greater than their individual behaviors. And kind of leading slightly on from that, I'm going to talk about how hierarchical design is this upward trend, almost in a kind of fractal way, but how these kind of vast patterns of life that we see and we know actually kind of originate from these small perturbations that win out over the dissipative effects of entropy, which kind of is the result of thermodynamics and those limitations. But we see that life is kind of perched on the edge of chaos. It's surfing this kind of wave just about avoiding being dissipated, but also kind of moving upwards and making new structures. And this is the last wish-washy bit, I'll promise. Biological systems are these kind of, they operate best as these deep hierarchies of agents that are in constant flux. So they're flexible, but actually highly ordered. And we see that from proteins in our cells to entire insect colonies, and now we've actually started to find super colonies around the world that themselves are kind of, those colonies are interacting. So we can think about this idea really of, taken from software engineering, but of modularity, but particularly in biology, of flexible modularity. So when you have these kind of highly malleable but also redundant arrangements. It allows parts to be easily swapped out or recombined. For example, if an organism loses a leg, it's still able to function, which our machines often can't do. But what we find is that this ability to kind of recombine parts allows a kind of ratcheting evolution that really propels things forward, because it's able to be lazy and reuse existing recipes in new ways. So, to give you the kind of really important example, proteins themselves are kind of organized on 4 different levels of complexity. So you take a string of amino acids, and it will fold into these kind of helices, and then also these sheets, and then that themselves will kind of fold into this 3D structure. And then proteins can also then, like building blocks, stick together into these complexes on a kind of quaternary structural scale. And that then forms the basis of important molecules like spider silk or collagen, which is the basis of things like hair. So something like collagen actually then itself is deeply nested in these kind of recurrent motifs like for bones. So we see that collagen forms fibrils, and then those form fibers, and then those form bundles that then form bone. And the same is true of things like bamboo, for tendons, and silk. So these are all kind of hierarchical structures. So now I'll try and get to a kind of a case study of some kind. So this one is on the ecological scale of nest building. So you can see nest as this additional interface or kind of buffer that an organism builds between it and its environment to make things a bit more cushy. And it's an interesting case study for biorobotics, I think, because it involves a lot of challenging movements, but also you have to deal constantly with very irregularly shaped materials. And that's why I think things like weave birds are so impressive. So this is a social group of nests that these birds will build, and they'll weave them out of grass just with their beaks. And clearly we're not on that level of kind of intelligence yet, or kind of robotics. But my favorite example is actually weave ants. So these are examples of agents that are able to use tools to actually adapt their environment. So they build these kind of leaf nests with silk kind of glued together, and actually the ants themselves can't produce silk, but their babies can. So what they do is they carry the larvae in their mouths, and they use them to squirt glue between the leaves to stick them together. But then they also kind of recruit others in this kind of very collective, coordinated effort to pull huge leaves together. And so you can see how this is a very kind of useful case study for kind of swarm robotics. And just to compare this to our kind of current robotic systems, they're much more easy to control, but they're incredibly rigid and inflexible. So they have a kind of gantry or a very fixed plate, or maybe they can move along a rail. But the reason why we make them so rigid is because we don't know how to control them in a more complex manner. And we're starting to kind of find new ways. This is a vine robot, this is a wall climbing robot, with things like soft and mobile robotics. But we're kind of just scratching the surface, and they're still very hard to control. We have to use things like reinforcement learning in order just to be able to make them move and walk. But they're actually much better at improvising, because if you think about it, for example, one of the legs gets knocked out, it can still walk. It still knows that it's got other legs that work like that. And so it's a very kind of different ability to kind of build structures. And that's why I got interested in spider mites. And for some reason the video is not playing, so I'll show you this next slide. I found these nests that were up to about 5 meters in size. And it turns out they were made by these mites that are about the size, they're about 1 millimeter in size. You can just about see maybe 1 or 2 in here. But they built nests that are about 5,000 times their size. And they feed on the plants, they're actually parasitic, and they're used as a natural form of pest control. But why I was interested in them is because unlike ants or bees, they're not actually a swarm insect, they're an arachnid. But they can work flexibly either by themselves, they'll still be able to aspire, but they can also work in huge colonies of thousands. So when I saw them, I was kind of amazed by them. But they build these structures that kind of cover plants in these dense silk mats. And what that does is it conserves humidity, which stops them from drying out, but it also protects their eggs from predators. And what we see is, especially because they're not like a swarm insect, they actually just selectively cooperate. So they're still competing for mates, but actually just because it helps them to have these kind of highways between plant leaves, they'll work together to solve this kind of inadvertently this optimization problem, which tries to kind of use the least material to cover the plant in this protective structure. And kind of one of my favorite architects is Otto, and it turns out he was kind of interested also in tensile structures. So he used things like soap bubbles perform kind of computer simulations around to build these structures that look just like that, actually. And then to go on a kind of level deeper, I know I'm saying a lot of information here, but I want to talk about tissue growth. So this is the other kind of study that I was doing earlier this year. So we see that cells coordinate, not just in sending each other kind of biochemical messages, but also by nudging each other with mechanical stress sensing. So they can, or rather with mechanical stress, they can sense kind of tugging and nudging forces. But more than that, as I was saying earlier on, cells other than neurons can also fire these kind of bioelectric potentials between each other. So I was looking at how leaves develop, but I'll show you this kind of slightly bigger example. This is phylotactic patterning. So it explains why we get these kind of predictable mathematical patterns, like the Fibonacci sequence, in certain leaves, and also things like these broccoli florets. But actually, this is leading on from Alan Turing's work. So it's amazing today that, 2018, you still have papers that are citing Alan Turing. But what he developed was this kind of model of reaction diffusion, which explains that as you get these are kind of a time lapse of chemical concentration of a particular kind of growth chemical called auxin. And what you find is that as this chemical propagates, you get these waves and maxima and minima. And whenever you get a maxima, which is just basically a lot of concentration of auxin, you get a leaf. That's a signal that says to the plant, please develop a leaf. Just right here. So this explains the kind of pattern. And what we see is that, talking about those kind of feedback loops I was talking about earlier on, is that actually this process is up to things like proteins inside the cells, which transport auxin. And as they transport auxin, it actually creates a positive feedback loop. So them binding to, or kind of interacting with auxin, means even more auxin flows. And then what we get is these kind of swirling, flowing patterns that give the leaf their shape. And this touches on something really important, both in animal and plant kind of development, which is that of tissue polarity. So here you see an embryo developing, and what it's kind of the green and red zones are showing different kind of the left and right side being kind of decided and developed. So my kind of analogy for this is like a stadium with all these different gates. The cells will organize amongst themselves which part of the tissue is going to be the head, the limb, the finger, the eyes. And if you kind of plot cells from one area to another, they'll change, they'll kind of adapt to fix what you've just kind of tried to disrupt. But it's a very kind of redundant or error-correcting mechanism that basically decides where things go, because you can see why that, if it goes wrong, causes huge problems for organisms. And this kind of, I can't talk about this stuff without talking about Michael Levin, who is kind of a heavyweight right now in this area. And he's growing two-headed worms. So he was looking at how, if you cut these worms in half, these are called planarian worms, they're highly regenerative, which is why they're being studied. If you cut them in half, usually they'll just grow either the tail they were missing or the head they were missing. So what you get is two new worms that are perfectly healthy. However, I was talking about earlier on about these kind of bioelectric signaling pathways, and if you actually mess with those, you tamper with that, then instead what you get is a worm, because you confuse the cells in what they think should go there, and so you get a worm with two heads that functions, or a worm with two tails. Why that's useful, we don't know, but it shows a very important effect, which is that tissue polarity is being done with things like biological functions. So really what I'm trying to drive home through these examples is that cells are intelligent. And this is what we call kind of basal cognition or basal intelligence. Because tissues then as a whole are capable of self-organizing, self-repairing, but also self-terminating, which is a very important and kind of neglected one, but it's knowing when to stop. Because when you don't know when to stop, that's exactly what happens with cancerous growth. And that's why cells usually have a kind of mechanism, they know when to die as well. And when that goes wrong, that's exactly what we see in cancer. But not only this, we have to kind of realize that some of our old models were wrong. That cells are more flexible in function than we thought. They're very good at kind of improvisation. So the old model we used to have is that cells kind of trickle down this landscape from stem cell, which is the kind of most generic form of cell, in order to, into these kind of particular cell types, like neurons, epithelial cells, and also like things like liver cells. What we find is that certain cell types actually can be remodeled. If we can kind of talk in the right programming language, we can fit the right genetic switches or regulatory switches, we can actually get the cells to change type completely. And this is kind of driving home that idea of flexible modularity. So the parts aren't just kind of fixed and rigid, they're actually a bit kind of pliable or compliant. And the kind of last bit I'll talk about is on the really, really small scale, and that's at proteins and organelles. So this is the stuff inside your cells. So we kind of think about these kind of collective interactions. So in things like protein folding, you've got loads of atoms involved, but actually these kind of strings of amino acids fold in predictable shapes. But the way it does this is not by using these kind of rigid nuts and bolts, or kind of strong chemical bonds. It actually uses these kind of very weak, non-specific interactions. And what that does is it gives it some kind of flexibility. This means that it can kind of self-organize or self-assemble to its shape with very little energy, because what it's doing is just thermodynamically trying to get to the lowest energy state. And this is a really key principle for self-assembly. I think this will help kind of explain it slightly better, more intuitively. But if you think about Velcro, it's a series of tiny, like lots and lots of small, tiny hooks, and it's very easy if we, try and pull it apart to just rip it. But actually, when you pull it this way, it doesn't shear easily. And this kind of idea of using lots of weak bonds rather than one strong bond is allow us to have a lot more kind of flexibility for things like shoes. But on the kind of DNA level, we see the same thing actually. The DNA is a very stable molecule under the right conditions, but actually these kind of hydrogen bonds are very weak between the two kind of different base pairs. And what that means is under certain conditions, when we're trying to kind of replicate DNA, it will completely unzip quite readily. And this is kind of what leads to metastability, which is the property of kind of being just about able to hold together, but can also be taken apart. And this is exactly what happens, a kind of similar effect in microtubules. So when I was talking about that kind of inner skeleton of the cell, it's made up of these tubes called microtubules, which are actually just made up of lots of protein building blocks themselves. But what happens is that these tubes are constantly being built up and broken down, built up and broken down constantly. And what that means, especially kind of on a small scale, is that they can kind of really probe for the optimal configuration. So actually, even in plants, in the space of half an hour, this kind of blew my mind, the entire scaffolding inside a cell can completely remodel. So plants that we think are these kind of static, boring, lifeless objects actually are completely remodeling the entire time, and it's essential to their function. So although it's maybe kind of questionable whether this is intelligent, this ability to kind of use things that are kind of dynamically unstable, that are being built up and broken down, but they will settle in a kind of useful state, that's the selective stability part, means that we can do some very useful and important computation there. So actually these kind of tubes not only provide structural support, but on them walk these proteins that carry important building blocks to where they're needed in the cell. So in plant cell growth, what they're actually doing, a bit like a builder carrying bricks, they will carry important construction material to the edge of the cell, to the cell wall at the weakest points. And that's because these networks respond to mechanical stresses. So they deliver material exactly where it needs to go, because the kind of cytoskeleton is aligned with what's kind of weakest. So what we see really in things like plants is that these microtubules will also arrange in a kind of band that kind of squeezes the cell and provides this force that only allows the cell to expand or elongate in one way. And it turns out that's about the whole mechanism, well, that's the most important part of the mechanism that gives leaves their shape. So these kind of tiny tubes made out of proteins that are kind of ranging in a particular way act like a compass, and they choose which way the leaf stretches. And that's a very important part of development. So we see from kind of subcellular effects that actually you get these kind of huge coherent flow patterns. And that's kind of mind-blowing to me, that basically tiny effects have can kind of cause these entire shape changes. But it's a very tightly controlled process of cell division and elongation. So that's the end of the information. That was a lot, but I'll just conclude. So if we think back to the kind of future of architecture with all of this in mind, all of these principles from biology, we can see building really as the next logical kind of level really in biological kind of evolution, is that we really ought to kind of think about buildings as these living organisms. Because we only currently design buildings to last about 50 years. Whereas some of nature's recipes, as I was kind of outlining, can last over 3,000 years, but not only that, they're biodegradable as well. So what if we could grow entire buildings using these kind of principles? And with that comes this kind of paradigm shift of designing, instead of going for this kind of final form, it's one of those like, it's not the destination, it's all about the journey kind of thing, but it's about designing for assembly and disassembly. So what if we focus on the algorithms for growth and for improvisation rather than kind of going from final form? And that means we have to have less reliance on kind of these homogeneous parts and kind of very rigid fastening mechanisms like nuts and bolts, and work with the kind of actual material properties itself. So this is one of my favorite architectural projects that uses the natural kind of curvature of bent bolts that are otherwise not useful for building construction or for making planks, and use them to make this kind of structure. But again, it's a lot harder to model that. So I ended with this idea that flexible machines make really great teams. So what if a bit like cells, we could devise these kind of soft robotic building blocks that could grow but also repair and remodel to suit the changing needs that we have for them. And then, if you think about it, then we can also use these kind of cooperative agents as part of teams that could adapt and improvise to solve different tasks. And we have this kind of idea of flexible modularity, where if they're kind of able to perform different functions and improvise, then actually they can achieve much more kind of complex functions. So with that, you end up getting a consortium of agents that hopefully, under the right kind of setup, will synchronize and they can reach the consensus. And that means that we can land on these kind of crowdsourced solutions even better than ones we could have predicted. So with that, I hope you enjoy the exhibition. Please take a look around. The bar is also open. But also, I just wanted to say I'm going to be launching my kind of art science project, which I'm really excited about this year, called Material X Algorithm. And hopefully that means this exhibition cascades this summer, which is all about everything I've just talked about. But yeah, I'll leave it at the quote. Yeah, so thank you very much. It's the cells of the base belief that are doing kind of dividing at the later stages. So really what's happening is that the cells on the edges are getting pushed outwards. They've already kind of done their job. Because otherwise you have to organize that and make sure they don't kind of divide out of control.
Right.
So it's a clever mechanism where you kind of work from the outside in.
Oh, outside in, yes, yes.
But what defines when it stops? Or maybe it doesn't stop.
It's usually to do with you switch from particular kind of chemical regulators that are telling it to expand and divide to then other ones take over. This purely genetic kind of groups, regulatory groups.
So like you have lots of kind of distributed compute to build these large-scale structures. Is there any sort of, within that, do they have an idea of, obviously they're not cognizant themselves, but is there an idea of where they are in the global system, or are they just going off of their neighbors?
So that's what's really clever, is that there's no way they could have some kind of global position. That would be kind of the top-down view. So when I was doing a kind of project where I was teaching cells and machine learning to grow something like a leaf, what you're trying to teach them actually is the rules for successful collaboration. So what they're doing is they're using their neighbors to kind of propagate these waves or signals outwards that mean they don't need to know exactly where they are, but just based on what their neighbors are doing, they know that they're in the right place and they're doing the right thing. So it's a clever kind of distributed algorithm. It kind of goes against our intuitions of knowing where, because otherwise you have to store in the contracted code also a way for them to find their position and to know what, at that position they should do.
I was just thinking, I was just amazed by the amount of intelligence and information and analogies. And then it came down, I did not know where am I coming for and what is it. But then eventually when it came down to the implications of it to architecture, I was like, damn, the amount of this knowledge you could heal the world, bring world peace and heal all the diseases. And then we build a building.
Yeah, I mean, hopefully we can, there's a bit of a step between that and like, I think what I still feel like is some of this stuff feels quite abstract. So I'd love to take it to more like a stage where we could reuse it, in architecture today. I think it does feel like though there's this wealth of knowledge from biology that we're not using. And it's exciting to me. I think it's a bit like right now. I think with things like AI, you can feel like things are all figured out, but actually this shows us that we have a lot more to learn.
Really,
Thank you. Go for it.
Yeah, I'm sure this is a very strange sort of question, but if you think of a caterpillar pupating and then becoming an adult and changing from being a sort of being that's programmed to eat to a being that's programmed to reproduce. Yeah, what would you have to say about whether the intelligence in that process of sort of pupation, when in a way the whole being is sort of disintegrated into its component parts, and then it's sort of reassembled to become a butterfly or moth? Yeah, where is the intelligence?
So caterpillars, I don't know enough about them. I looked into them a while back for this exact purpose. They are super interesting. I mean, I think what you find is they do kind of shed, I don't know if it's an exoskeleton, but they shed some kind of structure when they metamorphosize. And what you find is that I think there's something about how they forget the old function because it gets taken over by other things. The only analogy I can think of that I think might hold up there is that, for example, if a human loses their vision, we find that that area of the brain can then be used for other purposes and repurposed. So I think there's some intelligent mechanisms for cells that had a particular function before to be repurposed, but how exactly they kind of switch from having behaviors that are caterpillar munching leaves to butterfly flying is amazing. I don't. Yeah.
Yeah, I was going to use the example of the mites and the sort of arachnids building that net. How do you think it is that those tiny sort of not necessarily sentient beings under the traditional sense are able to solve that optimization problem, but humans that one would argue are sort of more intelligent, at least under the traditional sense, can't solve issues like climate change that are sort of optimization issues in themselves?
Yeah. I mean, yeah, that's a big question. I would say I ban myself from saying consciousness in this talk, so I won't address the thing of whether they're sentient or not. But I think the thing about consensus is really interesting because I know, for example, with things like ants, the reason why they're able to work together so well is because they're less self-directed. They're more cooperative and they're more about kind of, they'll put the swarm first. So, for example, they'll decide whether they're worth saving after an attack, whereas I think we'd be a bit more selfish maybe. And I think it might be that because we have stronger kind of independent goals, maybe it makes it harder to reach consensus. As in, it's harder for humans, especially en masse, especially with polarized views, to really come together as predictably as they do. I would think that because there's stronger coupling between them, or rather there isn't even a better communication between them, they have less communication ability as far as we know, but they're definitely less kind of self-directed. I don't know, yeah, that's a good question. Yeah.
I was just thinking about, you talked a lot about like molecular biology, and I was just wondering, you made like a lot of parallels between that and the kind of principles of computer science. So I was wondering, when you're kind of thinking about swarm behavior in robotics, and kind of, you mentioned the example of DNA methylation, of like not changing the genetic code necessarily, but it is going to impact like downstream when you do like translation of proteins. So do you kind of see an application where you, in robotics, for example, kind of changing and adapting to different stimuli that can kind of feed back to how it's being computed and monitored, so that can change its behavior?
Yeah. So I mean, in a really abstract sense, the idea of like structural biology of proteins having a physical shape that can perform some kind of computation is interesting because, for example, things like grippers, they're often very hard to control. So what you can do is you can use material properties, like that switch I showed, to program in features so that you have a similar control circuit. Because all you do is, let's say, inflate it and it will clench down into it. So that's a really abstract idea you can take from biology. But the kind of more kind of important or kind of nuanced ideas about things like gene regulation and kind of that kind of stuff, I think that's where it's more interesting for things like bioinformatic computing. So both in terms of how to build more kind of not only different kinds of neural networks, but also things like reservoir computing. There are other kinds of models of machine learning that potentially, I think, those kinds of things could be used for, inspired by. Yeah, I would love to see more kind of stuff done with kind of physical objects, but I don't know how much of this is the thing I've been debating. Do those effects like hold up on the larger scale? So, for example, the way a protein folds because of thermodynamics, could you still have self-assembly on a much bigger scale, or is that just limited by physical constraints? Because, you know, like, I guess electrostatic forces don't scale up in the same way. But I don't know, I was thinking about that. All right, I think I'll end there. Yeah, thank you very much.